I come from. I come from Loma Linda University at California, and we were the first one to do the open heart transplantation, and these are the kids given new heart by Dr. Leonard Bailey, and a couple of other things which are of prime importance in the United States. The foremost thing is that we have been classified under the blue zone, where people live in these blue zones are the ones who live more than 100 years, centurions. So if you live in California, we have seen a lot of blue zones. So technically to say, when you are a faculty there, your dean or your professor in head doesn't retire, unless and otherwise he or she dies. So I just put before you this lady who was the first centurion documented. Her name is Jean Calment. But the flip side is that she is a chronic smoker since 30 years. So there was a confusion here whether the genes played a very vital role or what. Here's another man, a very famous marathon runner in the United States. He started marathon running because his father passed away at the age of 35. So he wanted to beat the genetics. So he started running at the age of 25, and lo and behold, he died at the age of 37. So very confused, huh? Genetics or environment or epigenetics. So there is now a tangential shift where now you might have heard today, there were a lot of speakers who had set the path towards environment, microbiome, and even Dr. Anil was just talking about microbiome, which is more important. So having said that, talking about microbiome, we are all unique people with unique microbiome. And it all started with the Human Genome Project, and people are bored about Human Genome Project now. They have moved on to Human Microbiome Project since 10 years. But the main part is there are a lot of slew of data that have come out, but without any proper sequencing or without any proper analysis, we are not able to get any new data. So with this 10 trillion human cells in our body, you need to know that 100 trillion microbial cells do exist. So this is the attachment that we have with this microenvironment until we die. The microbiome initiatives towards the whole world, each and every country had taken its own initiative, but India is yet to venture into that, and you have heard from Dr. Rakesh, where CSIR is venturing into a 5,000 microbiome project. And DBT is actually floating a RFA soon. So people are venturing into microbiome, and microbiome work is pretty costly, but at the same time, the impact of microbiome is being very well heard now. So microbiome was given more importance since 10 years because people had related bacteria that bring about systemic diseases. And bacteria that is there in the food, they have a gut-brain axis, they can even change your mood. People focused on individual microbiomes and then ethnic microbiomes. There are a lot of studies that are going on. And one of the important microbiomes that we at Loma Linda study is the dynamic microbiome. We do the microbiome study with the marathon runners there. And there are a lot of good data that we got. And the other tangential shift was on the non-cultured or yet unculturable bacteria that we get during sequencing, which is of much importance. So this was actually published by one of our collaborators in 2015 in Nature, where you could see a plethora of viruses, bacteria and fungi, and out of which you could see the gut microbiome and the oral microbiome are the mainly diversified microbiomes. Okay, so what really happens here? See, when the baby is given birth, if it is going to be a normal vaginal delivery, the baby incurs the microbiome, vaginal microbiome from the mother. If it is going to be a C-section or a cesarean section, the skin microbiome takes its impetus into the child. But whereas this co-evolution takes place for a longer time, 1.5 billion years, they easily co-adapt. And ultimately, you could see that they are collectively now called as holobiomes. So it is not called as a microbiome now because it is a part and parcel of our body. So what we did was, this is the recent paper from us, where what we did was we actually collected the gut microbiome from all these continents. So the number of samples that we got was 13,313, with Europe 7,000 and odd, America somewhere 4,500 and odd, etc. So what we did was we did a whole genome sequencing, the percentage of gut database reads that we got by 100% with the locations that we found. And also we just categorized them with the health status, disease as well as the health status. And also we just categorized them into group where each got 50% of the reads good. But ultimately when you just see here, this is the human gut reference databases that do exist now. You could see here North America, Europe, Asia, and Oceania. They are all having a very proper sequential interactions that we could relate the bacteria or the microbes to the existing one in the database. But whereas you could see the uncharacterized metagenomic sequences that are there in green, which are very high in South America and in Africa. So what does this say is that there are certain pathogens that are unclassified right now. And I should say here Dr. Sivaji has even found 3 clusters of bacteria and which are very unique and that have not been nomenclatured at all. So we are just coming on to that kind of a path where we find a lot of these kind of data. And what we really find is that you could see here that we see a group of organisms that you see in the blue color, which is actinobacteria. The other one, the green one, is the bacteroids. So what you see in this area is the small red dots, which are exclusively found in the Europe and the North American population. But whereas you see certain unique ones that are in blue, where they are found everywhere on all the continents. So coming deeper into that, what kind of a bacteria do exist within these groups is majorly 2 separate kinds of bacteria. One is bacteroids that you would see here in this area. And the other one is fusobacteria that you could see here. But out of this fusobacteria, they are very less in number and they are not being characterized much. So this is another study where we found from these 3 different groups of people that we classified them fully. And what you would see here is we have just classified 12 different families, out of which these 12 different families are very unique to these 4 groups, ethnic groups. And the most important thing is we have even zeroed in on a couple of bacteria that belong to family Odoribacteriaceae and Odoribacter, what you see there, which actually their deficiency could cause Crohn's disease or ulcerative colitis, because they produce butyrates and they are directly proportional to preventing them. Coming to the vaginal microbiome, this is from one of our collaborators here. This is a study which is between Asia, white, black people and the Hispanic. You would see here, the group that you would see on the 4 is the lactobacilli group, where the lactobacilli group are more, but the specific lactobacilli, they cause probiotic effect or a very beneficial effect is very less in blacks as well as in the Hispanics. So these kind of health disparities do occur in the microbiome side rather than in the genetic side. So talking from the oral microbiome right through the rectum, the large intestinal microbiome, the microbial biomass moves from a smaller level to a very high level. But what is unique here is that you will identify that the ones that are there in the mouth are very static. Rather, the ones that are there in the small intestine and the gut are very mobilizable. Imagine they are being flushed out every time, and getting a microbiome settled is very harder. But whereas a microbiome that settles in your mouth, it is attached with your teeth, and the plaques that are being formed, even though you are going to brush, they are going to be staying there for a while. So this is the designated out of all these kind of microbes, 3.7 to 10 to the power of 30 microbes. See, the International Agency of Cancer Research had designated 5 microbes, out of which Bacteroides fragilis, Fusobacterium nucleatum, these 2 are bacteria that are present in the oral microbiome as well as in the intestinal microbiome, gut microbiome. So one good example of these 2 bacteria that predispose to cancer is that people who eat red meat. And if you're going to eat red meat, the carnitine and the choline, when they are just being metabolized by the gut bacteria, they produce trimethylamine oxide, which causes atherosclerosis. A very simple reason. I should say here that at Loma Linda, 95% of the people are vegetarians. We don't eat any meat. But for the CCMB terrace, we don't take liquor too. So I'm just telling you that this kind of red meat versus a vegetarian diet plays a very vital role, because this microenvironment has found a way in which a separate niche has been formed which is beneficial. Likewise, there is a term which is called dysbiosis, which is exactly the opposite of symbiosis in each and every niche. When this is being altered, then comes the problem where the metabolites, inflammatory cytokines, and pathogen-associated molecular patterns from the bacteria, they produce a plethora of diseases that you would encounter. Yes, genetics plays a very vital role. Ethnic diversity plays a very vital role. The diversity of microbes do play a vital role as well. So we are zeroing in. What you see in this picture is the diversified phyla and the genera that you see in the mouth. More than 900 different bacteria you could identify from the oral cavity. The next one is the intestine, out of which you would see here 68% is yet uncultivable bacteria. So human microbiome has been linked to systemic diseases more. A couple of bacteria have been very well documented that they produce or predispose systemic diseases: diabetes mellitus, Alzheimer's disease, rheumatic arthritis, atherosclerosis, stillbirth, cancer, and multiple sclerosis. So our lab is focusing on the microbiome initiative. And this human microbiome initiative, we are focusing on two important pathogens, which we call it as keystone pathogens. So the ones that you see in green, they are called keystone pathogens. Okay. So when you just see the disease status versus the health status, you would see there is a conserved microbial community that we have found. And out of this conserved microbial community, you find in the disease state, that is the red ones, we found these two keystone pathogens very well interactive to form a consortia. And we also found, we did a metabolomic study, and we also found that a couple of metabolic functions are very upregulated during these two bacteria causing disease condition. And what you just see here, this is the each and every pathway, and this is the sequence by which I'm just showing you the pathway and the bacteria that are involved in the microbiome in producing this variation. Where you would see here, Porphyromonas gingivalis plays a very vital role here. Wherever you see PG, you would see, here you see PG, PG, PG. So everywhere you see here, in every cycle, every pathway intervention, you would see this bacteria playing a very vital role. So why do we choose only these two bacteria? One of our cohort study, we also found that the bacteria that were in close coordination were Filifactor and Porphyromonas gingivalis. So when we started working on Filifactor allosis, this was called as yet uncultivable bacteria. It was very slow to grow, and we prepared a medium by which now they are growing it faster, and now you have more than 220 publications, and we have the seminal publications of this bacteria. So with this, what we did was we focused on these two pathogens. What you see here is the cover picture in infection immunity, where these two in 2014 and 2016 they came up. When you just see this electron micrograph, what you would see here is this is an infected cell, and you would see the vesicles here, and each vesicle has one rod-shaped and one cockeye-shaped bacteria, which is none other than Porphyromonas gingivalis and Filifactor allosis. So to say that if in a microbiome, if there is going to be a microbial consortia, that two detrimental bacteria could join, they could cause a havoc. And this is the main picture of an evidence that to show this. So we wanted to just delve in, and we wanted to know what is the variation that causes. So we studied both the pathogens as well as the host proteome. It's a global proteome analysis. And we also wanted to just focus on the pathogens. And when we focused on the pathogen, keystone pathogen proteome analysis, we found two enzymes to be highly upregulated: peptidyl arginine deaminase. Another one is Sialoglycosidases. The fascination about this peptidide arginine deaminase is that they cause citrullination of proteins. Citrullination is a process by which they cause rheumatoid arthritis, very plainly I should say. And these were the patients that have been taken from Loma Linda University with their permission. But whereas the other enzyme helps in the sugar code for exact attachment with the human cell. So we focused on these two because they are highly upregulated. So we're coming to the citrullinations part. What does it do? There are two main diseases that are caused by citrullinations. Number one is Alzheimer's disease. And there is a counterpart in the University of Florida who's working on Alzheimer's disease with this same bacteria. And he was all along in the news in ASM. And we are working on the citrullinations to rheumatoid arthritis part, where the citrullinated proteins by these bacteria, they produce antigen antibody complex, they settle in the neurons as well as in the joint to cause this disease. Coming to the host proteome now, we also saw a couple of host proteins to be highly upregulated too. So this is what I'm just showing you here is an Orbitrap analysis, a very extensive analysis crunching down. And we did an association network centralities pathway, where what we did was very simple, that we went into the PubMed, we just searched in the keywords where what are all the proteins that are highly upregulated in rheumatoid arthritis. And what we found was, we found these important proteins that are very highly upregulated and being identified in RA. And so to say that if you just match our proteomic data, you could see here they are all highly upregulated ones that have been identified in our this thing. So having said that, we went into further deeper to see whether deamination or citrullation does happen in each and every protein. And what you see, R is the arginine, and arginine gets deaminated, and what happens is a very big protein gets chunked up and they form specific antibodies, and the antigen antibody complex they join together and they get settled. So these are all the data to just back up that we did a slew of analysis with all the host proteins. We found deamination or citrullation on the arginines. Even one or two citrullations in a protein is more than enough to cause citrullation and breaking down. And so these are all important proteins I'm just showing up, where you could see this is R is arginine, which is citrullinated. And this analysis, we found that major proteins that to just pick up, they are citrullinated are the four important proteins that are highly involved with epigenetic modulation. The next story is on Sialoglycosidases. See, these two enzymes play a very vital role in the attachment by glycans. You know that the surface of the eukaryotic protein has the sialic acid, and each and every cell has unique sialic acid. And the interaction of this unique sialic acid to bacteria, the bacteria has to mimic and produce a similar counterpart. So the hypothesis here is that we wanted to see whether do we have a unique sugar code that affects the whole microbiome, by which each and every bacteria that causes very detrimental effect can use this sugar code in the attachment. So what you see here is the blubbing vesicles that you see here, and they contain the virulent factors, the cysteine proteases, etc. So the hypothesis is this, that the sialidases, they actually break down the terminal sialic acid, and ultimately they play a very vital role in host inhibitory signaling and also evasion of phagocytosis. It means to say that if this bacteria with this enzyme is there, the bacteria is going to be sustaining in your system for a long time, because it is going to evade the immune system. And any counterpart who is going to be there with this keystone pathogen is going to jolly well live along with this keystone pathogen. So they affect the Siglec-9, okay, the sialic acid binding lectins, which are very important immunological factors, by means of a Siglec ligand CD43. So we went very deep into that, because CD43 sialophorin is more important for this interaction to happen. So when you just see the electron micrograph picture, what you see here is a mutant of that particular sialidase gene, where you would see here there is the bacteria is just like that, but whereas when you just see here, there is a very strong covering that happens, which is very important to evade phagocytosis. And we also did the overexpression of this CD48, and we also found that there is an overexpression that you could see here that helps in the attachment. of this particular bacteria with a common sugar coat. So this is what you would see. Imagine if it is going to be a non-infectious bacteria, your human cell is not affected at all. But whereas if it is going to be an infective bacteria with two counterparts together, one is a Porphyromonas gingivalis, another one is Filifactoralosis, you would see that the whole cell becomes so rumbled. So we went in to just scoop away all the cytoplasmic material. We took only the skeletal part. We wanted to see what is the sugar coat. Likewise, we took the vesicles and we wanted to see what is happening to these vesicles. And we found a very important type secretion system, type 9 secretion. Type 8 secretion systems are there between bacteria and the host, but we found type 9 secretory system components very well. So we went further down to identify the glycans, the common sugar coat. So what you see here is a glycan reductive isotope labeling LCMS method, and what you see here is a standard, and this is also a standard of all the glycans that exist, and predominantly they exist in the interaction. And this is another performance to identify the individual disaccharides that form these glycans. So to say, what happened was, we took human neutrophils, we took P. gingivalis cell, we took P. gingivalis outer membrane vesicles, we also took the secretions of P. gingivalis. We found some unique sugar coats. The work is still underway. We found some unique sugar coats that are very, very essential for the host pathogen interaction. And I should say that Porphyromonas gingivalis and Filifactoralosis does exist in every ethnic group. And there is no ethnic group that is devoid of this particular thing. So we also just confirmed the expression of these glycans by the Porphyromonas gingivalis in the macrophages too. But the thing is, what will happen here? So we talk about microbiome, and which microbiome we think is the best one in the whole world. Because people can take the microbiome and they can go and take them as a probiotic thing. But this is one of our collaborators from Harvard. They took the gut microbiome from Hasta hunter tribes from Tanzania. And what you see here is the Tanzania tribe people who had the highest diversified, very good microbiome in the whole world. And now people are just venturing into getting that microbiome and making them as probiotics, where Loma Linda is one of the counterparts with these kind of things here. So having said that, oral microbiome leads to systemic diseases, and it's a proof of concept. We proved that there is a lot of relationship between oral hygiene and head and neck cancer. And butyrate, as I've told you, certain group of bacteria, the butyrate, play a very vital role in the bioavailability of the drugs that we give. And this is one of our publications where this said that oral and gut microbiome is perturbed in rheumatoid arthritis, where we have just presented this thing too. So thank you very much for your patient listening. This is my small group which is attached with the human microbiome work. And I really thank the NIH and IDCR for 2 grants which actually support this and still going on. My collaborators from Harvard, from Germany, these 2 are my students, and the other people. And also Dr. Kesavalu, who's working on Alzheimer's with Porphyromonas gingivalis. Thank you very much.
The top